And I'm really happy to talk in this summer workshop. It does surprise me, I mean, Shannon always looked forward, and all of you guys are sitting in the back half of the room. So I don't know what is special about this CSoI, Center for Science of Information. Maybe information has to travel a bit farther before we can get through. So I would like to tell you a little bit about applications to life sciences. I've been involved with the Center for Science of Information since before its inception. I was actively engaged in writing in fact, I distinctly remember the whole idea of the center started in San Diego when Wojtek and Anant were visiting me, and we sat in a coffee shop. We were talking about this thing, and we brainstormed about creating this center, and then 2 months later we were busy writing a proposal, and now we have the center. So therefore I'm really happy that we have 5 years to the center now, almost done. We are looking forward to the next 5 years. I mean, clearly all of you here understand the importance of the science of information with the influx of large volumes of data in every walk of life. All you need to do is to go to Google and search Google search. So you've got large number of things, large amounts of data, making sense out of it. Data to information to knowledge is the key towards understanding this. And so this is what I'll specifically talk today about life sciences. I mean, as you know that, Todd just mentioned, we have this $120 million project called Pediatric Genomics and Systems Medicine. The idea is to sequence every child who comes in, and then we'll at that point in time digitize. Essentially, it will be a digital representation of human existence and life. It's not that you get ill and you get digitized. You are digitized that you can monitor lifestyle as you go along. In fact, the next-gen sequencing, about a decade ago, there was a real revolution in terms of this technology for sequencing the genome, sequencing the transcriptome, and sequencing in general. And it's a game changer. It's a game changer because it really sped up things in a super Moore's law fashion. And so in fact, already in 2012, we had, this is the number of DNA sequencing base pairs per second. You can see that it's doubling every 5 months. Now it's actually doubling every month. So therefore, you can see it is really incredibly super Moore's law, and so therefore we are getting large volumes of data. So the question is, what do we do with all this data? In fact, about 2 years ago, Francis Collins, don't read this, I don't need you to read that. 2 years ago, Francis Collins, who is the director of NIH, said that he wants to sequence 1,000 human beings, 1,000 genomes, so that we can figure out all the possible polymorphisms that happen in the human sequence, so that we can decipher what is normal, what is diseased, and how we can go about doing things. So this is in fact one of the, very, very exciting things. Now we have sequenced more than 1,000. In fact, there have been papers published on 1,060 or something humans who have been sequenced at this point in time. The take-home message in the sequencing is that each person differs. Is this color bad? Can you see from the back? Yeah. Okay. Each person differs from the current reference genome by a loss of function mutations in about 250 to 300 genes. And we have roughly, give or take 1,000 genes, 25,000 genes, give or take a few thousand genes. And so each person differs from the current reference genome. The reference genome itself is made up of around 13 human beings. And so we differ by about a loss of function mutations in about 250 genes. Each of us is also heterozygous for 50 to 100 variants that cause inherited disorders. We may never see it in our lifetime, but we in principle have these inherited disorders. And the study found that individuals typically carry more than 2,500 non-synonymous variants. That means it codes for a different amino acid in a protein. So variants at parts of the genome that are conserved across species. In fact, if you take genes that are conserved across mouse, human, rat, pig, monkey, etc., we see about roughly 2,500 substitutions that are variants of the genome. In fact, when you look at how much genetic variation does an individual possess, this was a paper published in Nature a couple of years ago. In fact, you will see that I want you to focus only on one data, which is that compared to the human genome reference sequence, we have about synonymous SNPs of around 10,000, give or take a few thousand, non-synonymous SNPs about 8 to 10,000. And then there's some deletions and losses and all these kinds of things. This is kind of, this is a tiny, tiny, tiny drop in a big bucket, because you consider that our sequence is made up of 3 billion plus base pairs. So in 3 billion plus base pairs, what is 10,000? It's a very, very tiny fraction, right? So therefore, we really have, this is like looking not for a needle in a haystack, but a hay in a haystack. So I mean, this is truly a challenge for information science. It's not a big problem. In fact, a genetic testing of a European individual showed that this is one individual whose genome was sequenced. I mean, he had the following inherited conditions, and he had the following health risks, and he had the following drug response, and this is all from sequencing the genome, right? Look, this is becoming now more personal, more individualized. This is an individual human being, and you can calculate your risk at 33.9. This became so much interesting that the next slide will tell you why it's so interesting. In fact, clinical sequencing is now becoming common. Some of you who read popular news know that Angelina Jolie had a double mastectomy recently, because she found out that she was a very high risk based on her sequence for breast cancer. So she decided to beat the punch and then go ahead and have a double mastectomy. So it's in fact called the Angelina Jolie effect. And in fact, now it's becoming routine sequencing of clinical sequencing, or at least it will become routine in the next few years. All of us will be having our sequences. In fact, looking at this, several thousand tests have already been ordered for patients with the goal of establishing diagnosis of common, rare, clinically unrecognizable and puzzling disorders that are suspected to be genetic in origin. And in fact, what makes it so exciting is not so much that you have a variation in the genome sequence, but you can have a variation in the regulatory genome sequence, which really can cause changes which we don't even know what is happening at this point in time. So it's really a large challenge. So you can tell me this is all biology. Why do I care? I'm an information theorist. So this is what is the problem. The problem is, what are the information theory challenges for this specific sequence-based biology or one-dimensional biology? The source, think about a canonical Shannon view. You have a source. The source in this case is species and inheritance from species. The encoder is the genome. The channel is the sequencer that we use to sequence. The decoder is the analyzer that we analyze the data from. And in fact, the complexity is species have functional relationships which vary. Genomes within species have conservation and variance. Within each species, there's significant variance, which is it's not noise, it is just standard variance. And then the sequencer can add noise. When we do a sequencing, we can do only within a certain range, so the sequencer can add noise to this problem. And then the analyzer is intrinsically statistical, because we use statistical methods, and so therefore there is uncertainty in that as well. So this is a typical canonical Shannon problem in terms of figuring out methods to figure out each of these cases, decipher the variance, look at the disease relevance, look at the risk factors and so forth. And this is a very typical problem in information theory. In fact, I believe people in Stanford are working on this problem of trying to figure out, are we going to hear talks from Stanford on this issue on the sequence variation? I don't think so. Not this school. Okay. So there is a significant amount of investment in terms of doing this. And for students who are taking this workshop, please stop me at any time. I don't need to go through all my slides. I'm happy to stop and answer questions or engage in discussions with you. Okay, now we want to go beyond the one-dimensional sequence, because most biology today is the level of excitement in biology is, there is a lot of excitement in the sequence. Well, don't get me wrong, but we have to go beyond this as well. In fact, our funding that we got recently, this large gift from a donor, was not just only for sequencing, but going beyond the sequencing to understand the phenotype. It is called the genotype-phenotype relationship, which means, do you have it? Does the child, is he or she likely to have a disease? What are the risk factors, and so on and so forth. So if you really deconvolute this, there are roughly about 200 cell types in the human body, things like the neurons, hepatocytes, cells in the blood, and so on and so forth. There are about 200 different kinds of cells in the human body: brain, liver, bone, heart, and many other structures. But the fact they must be reading of different sets of hereditary instructions written into the DNA. The fact that they can think about it for a moment from a purely information theory point of view, one coded blueprint, but they're able to read off, specific to each cell type, a different coded set of instructions. right? It's like having a set of books and each book having a different grammar, different parsing, etc. So this is really, the system is something like a play in which all actors have the same script but are assigned to different parts and are blocked from even seeing anyone else's lines. You cannot see anyone else's lines because you can get confused, right? So this is the real problem. The fertilized egg, if you take the egg, it possesses the first copy of the script. It's only one copy, it's the initial copy of the script, but as it divides repeatedly into 37.2 trillion cells, this is how many we have in our body. I mean 37.2 trillion cells, a few million cells die every minute and a few million cells are reproduced every minute. And the cells assign themselves to different roles they will play throughout an individual's lifetime. Somehow the cells get instructions from one cell to know what to do or what tissue and how to behave and so forth. It's a very interesting informatics problem. We haven't figured it out, all of it. People started figuring out the very first parts of it. For example, when you take the egg, the very first, an egg and the sperm, the very first thing that happens is you form germ layers. You form 3 different germ layers, and these germ layers start dividing and, start giving rise to individual lineages through signals. They have got various signals. Now, in fact, in my lab, we have worked on these initial germ layers. A student in my lab, Vipul Bhargav, in collaboration with other people, has worked on figuring out how to look at these 3 germ layers. We have the parts list, but this is experiment. We have the parts list for the germ layers. But this is not sufficient. This only gives you a starting point, and in fact what determines how you have these things specialize is called epigenetics. In fact, epigenetics determines the fate to some extent of the factors around. It tells you how these cells become heart cells, brain cells, blood cells, and so on and so forth. But what we can measure, we can't measure this yet. In fact, we have just started to measure epigenetics. So these measurements are really very early stages. It is not very robust, we can't measure very well. So we don't know what are the instructions or how the instructions are set. We're starting to figure out by using a recipe book. We are saying, okay, we add these factors, will it differentiate into these cells and so forth. But it's still too early in the game for us to have enough data for building knowledge. But what we can measure is the gene products of each cell type under different conditions. So what we can say is we can take your blood cell, your heart cell, your brain cell, your liver cell. We can measure the genes in those cells under any given condition. That will also change. But for a given condition, we can measure the genes. And this is called expression profiling and transcriptomics. This data is now enormous. It's really enormous amounts of data. It's all in public repositories. This has helped us retroactively identify cells and their function. For example, if I don't know what cell it is, if I do a gene expression, I can say, oh, based on these markers, this is going to be a given type of cell like in a muscle cell. So I will be able to do that. In fact, Anant Grama, who will talk to you at 11 a.m. today, he's going to tell you about how his student working with him and me had figured out how to look at tissues and how to look at tissue-specific identification of genes. And so we'll look at that later on today, in the talk. In fact, if you look at tissue-specific proteome, there's a paper that's published in 2013. I'm just giving you an idea. So if you look at each cell type, for example, an embryonic tissue or salivary gland or pancreas or lung or ovary, you can say they're all from the heat map. They're all different proteins or different genes. This is just a microcosm of what happens in the whole cell. It's just a tiny microcosm. So it tells you already that everything, some are more similar, some are more different. And so this is the kind of thing we need to figure out what is the function. In fact, one of my postdocs in my lab, he worked on this idea in a muscle, skeletal muscle, and for one given function, he can figure out, for example, this is a muscle cell. You can see a muscle cell expresses a number of genes, not expressed by other cell types. It's expressed only in cardiac muscle to some extent and so forth. So you can start figuring out which cells do which function. But all of this requires data mining, statistical analysis tools, and information theory-based approaches, which is what I will tell you for the next half an hour. So if you want to do this correctly, you need to know the parts list. So what do you do if you want to go to your home and find out everything that you have? You take an inventory of things. It's easier said than done. But we need to take an inventory of things. In fact, we published a review many years ago where we showed how many cell types are there, how many genes are there, and what type of things you need in our inventory. All of this, ultimately, the function happens because proteins talk to other proteins talk to genes, metabolites are changed, proteins talk to metabolites. So this is a constant flux of network of activity inside a cell. So what are our informatics challenges? Our informatics challenges are we need to take data and we need to convert data into networks that are functional. This is a typical reverse engineering problem. It's a reverse engineering problem because you want to take data, it happens because it's a function, but you are really reading off the parts list or the components. And so you want to say, I want to go back from this measured, observed data back to the networks. So this is the first challenge, from data to networks. In fact, in systems biology today, network reconstruction is one of the largest challenges that people face. And it's not in a context-free manner. You have to do it in a contextual manner, which means that a muscle network is different from a liver cell network, is different from a neuronal network. And so every cell has a different type of network. So you have to have semantics, you have to have information structure and context. And so this is fundamentally an interesting area of information theory. But this is only a static way of thinking about things. But cells are dynamic entities. In fact, your physiological function is a dynamic process. So how do you deal with dynamic process? So you need time varying data. That means you need to have snapshots at different time points. It's not just at one given time point. You want to have a movie of what is happening. And so in fact, in space and time, we need to look at dynamical networks and topology of dynamical networks. Shannon did not deal with this because Shannon dealt really with the static data and point-to-point communication. And so this is really where life becomes very interesting and very exciting for information theorists. When I talk to my information theory colleagues in the electrical engineering department, they are very excited about this because they say that this really will push the frontier of Shannon's approach towards looking at time varying signals. The last part is, per challenge, at least the ones I'm going to talk about, is that when you build these large networks, let's say I have a magic wand, I wave the wand, I take the data and build large networks. What does it mean? I want to convert it into knowledge in terms of saying that the network, this part of the network does this part of the network does this part of the network is involved in so and so function, this is normal function, this is pathological function. For that, I need to understand cliques in networks. I need to get modularity from large graphs. I need to look at big graphs and look at what modules are there, what function do these modules have, are the modules the same in two different contexts, are they same in two different human beings, how do we deal with it? So this area, in fact, is a very robust area of research in graph theory. In fact, it's called community detection, modularity building, subgraph building, subgraph isomorphisms, polymorphisms, how do you match those? This is a really hard problem. In fact, some of it is an NP hard problem because you cannot match subgraphs to other subgraphs and see if they are identical or not. So I'll tell you some work from my lab in all of these areas towards building networks. I'll pause for a minute. Is there any question at this point, or is it clear, or is it not clear, especially for the students who are in the audience? Does it make sense to you? Do the challenges make sense to you? Okay, so this is really where I think the future is going in information theory, in biology. You can apply it to communication networks, you can apply it to astrophysical data, what have you. But in life sciences, these are types of challenges which are very typical. So let me talk first about reconstruction of networks, data to networks. There are many ways of building data to networks. There are algebraic approaches to building data to networks, which I'll show you in a minute. There are methods which are statistical methods like Bayesian methods or other types of statistical methods, correlation-based methods to build networks. There are even information theory-based networks, which I consider under the statistical umbrella, to build networks. I'll give you an examples from my lab of various types of networks. In fact, the first one is data-driven reconstruction for linear and nonlinear models. Most of the times we think of these networks, most people who do network reconstruction think of it as a linear problem. Sometimes it is not a linear problem, it's a nonlinear problem. I'll tell you about linear and nonlinear. This is work by a graduate student in my lab who was supported by Center for Science of Information, Berang Asadi, and Mano Maurya, who's in the audience, who was partially supported by this grant. And in fact, they worked very hard on developing methods for building reconstructing networks. In fact, we published several manuscripts. I'm going to make this available to the students so they can really look at these manuscripts. I'm not going to have time to go into details with all of these for this particular type of these approaches. The basic idea is you have to derive, this is what happens in a cell. For example, if I were to be Maxwell's demon looking at a cell, and I'm going to look at the cell. Cell, for example, has a communication, has a signal. the signal activates this guy, this activates this guy, and this so on and so forth. And so then finally in a bacterial cell a motor rotates, and then the flagella goes out, and then the cell starts moving. So this is kind of a cartoon diagram of what biologists think happens. What we have is essentially, this is a network, this is essentially a network of interactions, and what we have measured is a set of data. If we have these experimental measurements, this network, when we measure this, we get things like this, and what we really want is reverse engineering, which is we want to go from this data back to the networks. So this is the basic idea. So you want to measure at, I mean, I'm not even talking about time, I'm talking about one snapshot. You want to go back into this network. So the basic main methods people use are linear regression methods. Here X is your input data, Y is your output data. If you measure your input and outputs, and then your coefficient, your B matrix, it gives you the set of coefficients. Essentially it gives you the connectivity, it gives you all the edges. That's a very simple way of thinking about it. It's a linear mapping problem. Okay? And again, I don't go into all the math details, you can read the papers. Most coefficients are non-zero, which is an artifact of the way we are mathematical artifact. So does it mean all edges exist? Of course not. No, not all edges are significant. If every edge is significant, then you have everything connected to everything else, and that is not how cells function. So you want to have some way of testing it, so you do perform statistical testing, and the statistical testing decides which edge is above the threshold of the statistical test, and then you say, okay, I'm going to connect that. Which edge is not, I'm going to ignore that. So that's how you build a graph, a model from your matrix model. And so, so that's the basic idea. But what about these inputs? These inputs are not all, I mean, let me go back here for a second. These inputs X are not all independent inputs. They're all linearly independent or completely independent inputs. Then you don't have a problem, because you can say they're all independent, so I'll be able to use my linear formalism that I showed you. They are, of course, they are coupled, inputs, which means they're correlated. So you have to transform it to figure out what is correlated, and then you have to convert it into a space, which is proper space, do the same process again. So in fact, people in many, there are many papers in many fields which do these kinds of things. It's not unique to biology, not unique to us. Okay, then there's methods which have been developed by people. For example, in Stanford, people developed this method called lasso. What lasso does is to integrate all of these things in terms of building the statistical, building your regression and statistical methods, interact them together. And so this is actually a good method, that's been used by people. The motivation we developed Berang in my lab developed, along with Mano, developed a new method where he integrated, he took the best of all these methods, matched them together or meshed them together, and then built a method which is called a DP lasso, which is a doubly penalized lasso. And the basic idea here is you take a model, which is your original linear model, you build a PLS, which tells you which coefficients are meaningful, and then you use the lasso procedure to do the testing, and then you reconstruct the network. So this is, we tested this method on a number of examples. We published papers in this area. In fact, we tested it on the yeast cell cycle. But it's a nonlinear model. What we proposed earlier is a linear model, but you need a nonlinear model. So therefore, for a nonlinear model, this is in this case, for example, as you can see, many edges are, this is our result, so many edges are overfit and so forth. So you need a nonlinear model. So nonlinearity is an intrinsic principle in biological systems. If you take a simple differential equation, kinetics model, it is a nonlinear model. So you need to worry about nonlinearity. How do we deal with this? You take a model which has linear and nonlinear components, and then you transform it into a linearized that model, and you can once you linearize that model, you can use the linearized model now to solve your expressions. Essentially your matrix becomes like this. This is your matrix. You combine your these two matrices, and then you essentially convert it into a linearized model, and then you can solve the linearized model. People in my lab have worked on this. And then our approach is, then it's the same approach as before. You have this model definition, lasso, significance testing, and reconstructed network. We can show, in fact, by using this hybrid linear nonlinear model, we can get substantially sensitive approach, high specificity, and high accuracy. So in fact, linear model, as you can see, does not do as well. So in fact, this is the kind of thing that people will use in the, towards building nonlinear systems. There's a student in my lab who was also supported by SOI, who worked on information theory based networks, and she graduated the master's degree, and along with Mano Maurya, who is here, She used information theory approaches towards building network reconstruction. The basic idea is, instead of using the algebraic approach towards determining the coefficients for finding the edges, you ask the mutual information, and you ask the question which mutual information is is meaningful above a threshold, and then you build a network based on the mutual information based approach. In fact, there's already an algorithm developed by Califano and co-workers called ARACNe, which uses the idea of reconstruction of accurate cellular networks using information theory approaches. Farzana and Maurya modified this approach, and then they used it to a complex network. Again, I don't want to go into the details of these because this is already present in papers. And then we can build networks like these. These are networks involved in, for example, from data involving in macrophages when you have a receptor and the receptor ligands are activated, then you build a network. In fact, you can have a combined large network you can build like this. These are the types of things which give insights to biologists to do more experiments. Again, my idea is not to make you read every single node here, but the idea is to tell you these types of networks allow you to do more experiments in biology and tell you what are the hypotheses. For example, if I have a drug, how will this network change? It will be a very interesting question to ask. These are the types of things we can address using these approaches. I'm going to skip this slide now. What about time varying? Everything I told you now is canonical, static reference network. What if it varies with time? Because everything changes with time. So we have to deal with causality and time varying network. Mariam Masnardi, who's a current student in my lab, supported by STC, she's, along with Mano Maurya again, who is very actively engaged in all these projects. She used an idea that was actually, helped by Todd Coleman for us to build in terms of time varying causal networks. So we just published a paper where the idea is that, let me skip this, we used the idea of what is called Granger causality inspired by Todd's work, and the goal here is that if signal X, let me go to the next slide, I'll show this better. If we say X is causing Y, if you are better able to predict the future of Y using all available information than if you use X alone. That is, if you have X, if your future of Y is dependent not only on the past of Y, but also on the past of X. It's a very simple way of thinking about it. And so Granger was the first guy to do this. Granger is from San Diego, in fact, in 1969, introduced this in econometrics, went on to win a Nobel Prize for this. And so this was adapted by people into, for example, in neuroscience. Todd did some work in neuroscience. We adapted it to building causality in networks. So formally, you can say a time series is said to be Granger causal at time series yt. The future of yt can be predicted given the past values of yt and xt, and you can extend it to all of xt. In fact, Mariam used a method called vector autoregressive model. I won't go into details here. The basic idea is to consider this dependence, and then she used this idea to build the same data set I showed you before in macrophage cells. She used the inputs and outputs in the cell, and then this is your correlation network. But if you convert it into causal networks, this varies with time. As you can see that it varies A, B, and C, and so forth. So we can now start building. In fact, Mariam just walked in. I was talking about her work. So we can start building causal networks in terms of looking at time-dependent data sets. This is very, very important in life sciences because nothing is static. In fact, all of us are dynamic entities, even though even if you sit and watch TV like a couch potato, you're still a dynamic entity because things are happening inside your body, in your brain, in your body, and so on and so forth. So therefore, we need to have a sense of dynamics. This is what we are going towards. In fact, we are continuing to work on building many, many methods which deal with causality at this level. The last part, how much time do I have? I don't have a watch on me. 10 minutes? 20 minutes. 20 minutes, excellent. So this is great. Okay, so we wanted to work on, I told you in the beginning about 3 problems. I told you in terms of building with using information theory based approaches. The first one was reconstructing networks. The second one was dynamical networks. How do you deal with time and causality? And the third one is how do we take large networks and identify cliques from large networks and try to figure out what is functionally meaningful, what is not functionally meaningful. So this is really another very challenging problem in this. It's the equivalent synonymous to taking very large graphs and identifying communities in graphs which are relevant. In fact, for those of you who would follow the internet, this is a very common thing. In fact, this is a highly invested area because companies want to know what you're thinking based on communities. For example, they broadcast you, they say, I mean, or webcast to you, I mean, all the commercials that are specific to you. I mean, or you meaning you not as individual, but you as a class of people who look at only Gucci leather belts or what have you. So therefore, this is a very important area in life sciences. So this work is, I'm going to tell you about work done by a graduate student in my lab, Tejaswini, who was supported by NSF, the Science of Information grant, and was we collaborated with Anant Grama, who will be giving a third talk today, and Meryl Gersten, a postdoc in my lab. So we have several papers that we have published in this area. Again, I'll make it available to the students so they can look at these papers if they are interested more in it. The basic goal is communities or groups of nodes in a network which are more similar to each other. I mean, I don't know how to define, you need to figure out how to define similarity. This is also an issue. But you want to really figure out, for example, you want to all the blues, all the yellows, all the greens, etc. So you need to figure out, I mean, these kinds of networks. A typical social interaction network subset is about 7 million nodes. So this is what Google does all the time, or Facebook, or what have you, does in terms of trying to figure out of the 7 million nodes, so what cliques are there, so they can send you commercials for British Airways or what have you. So this is really this modularity or identification of communities is an important problem in network theory. There are many algorithms for community detection. There are graph theory based algorithms. There are statistical algorithms. In graph theory algorithms, you can have agglomerative algorithms or divisive algorithms. Either you build from the bottom up, or you divide from the top down, or you have statistical algorithms where you infer a latent variable and build classification models. In fact, there are many, many approaches for building classification models today. Again, much of this knowledge is coming from non-life science community. It is, we are borrowing it in life science community, but importing it in life science community offers unique challenges which are not present necessarily in other communities. So this is really one of the issues we deal with. For example, semantics and context become a very important issue in life sciences. In social networks, it is less of an issue because you are likely to be more consistent within a time frame. You are not going to radically change from by being a Walmart customer to being a Nordstrom customer. So therefore, you are going to have more commonality from that point of view. Life biology is much more different from this point of view. So in terms of algorithms, agglomerative algorithms, you build bottom up. In terms of divisive algorithms, you remove edges, and edges are removed between least similar pairs of nodes, and you start building networks. In fact, the algorithm that Tejaswini worked on was called Newman-Girvan algorithm. It's an algorithm that uses edge betweenness to drive divisive approach. It uses a divisive approach towards community detection, and you expect the betweenness to be largest for intercommunity edges, and so paths between intercommunity nodes must pass through fewer intercommunity edges. And so the general idea of this algorithm is to calculate betweenness scores, find the edge with the highest score, recalculate between all the other edges, and repeat this process ad nauseum till you reach a certain point of convergence. So in fact, Tejaswini and Meryl worked on this convergence criteria. They used a geometric method for convergence, and I'm going to skip these slides. Again, these are present, and they used a geometric mean algorithm. They developed a geometric mean approach, and based on this, they applied it to a number of different protein interaction networks. And again, this is published, and so I'm not going to tell you about it. I'm going to tell you about one example of this, which has really been very exciting for us. In fact, this is a published paper. One of the reviewers said that this paper could open a new horizon for targeted drug discovery studies in dystrophy, muscular dystrophy. The paper is transformative, etc. I don't think it's that good, but anyway, the reviewers seem to think so, so we are happy about it. And so in any case, the idea of it was very good. The idea of it is you take, let me tell you first before we go on to this. We have large amounts of expression data from human skeletal muscle in normal patients and deceased patients. Disease in this case is muscular dystrophy. I mean, I don't know how many of you know about muscular dystrophy. Children are, I mean, at a certain age of a child, you find out the child has muscular dystrophy. Their lifespan is given. They don't live more than 16 or 18 years. They die, and the muscles essentially become dystrophic, and they have a genetic mutation which causes this to a large extent, but there are other things that are involved in terms of how this transforms, the mutation transforms into function. So one of our goals was to identify, based on this data from muscle in humans, I mean, we had actually several hundreds of humans at that time, And so we wanted to use this data from humans in order to ask a simple question, what is different between normal and diseased humans, and so that we can start looking at cliques. And these cliques could represent functions at various levels. So that's the basic idea. So in fact, when we looked at a data set, normal muscle, number of probes were around 7,685. We took only those experiments done in the same platform, same type of experiments, etc. And you can obtain this Newman-Girvan using the Newman-Girvan method and using the geometric criteria. We can look at differential analysis. We convert them into functional pathways. In biology, there's a rich knowledge. Once you identify a clique, you know the genes. If you know the genes, you can say these genes correspond to this particular type of function, etc. So in fact, we use this to look at, Tejaswini showed that in metabolism there is significant difference between normal and DMD. But more importantly, she was able to show the connections between how metabolism influences, for example, regulation of cytoskeleton, which involves motor activity of the muscle. So these kind of connections give you very interesting insights into muscle behavior in normal and pathological cases. I'm not going to go through this slide. In fact, she looked at metabolic pathways, coupling between pathways and actin cytoskeleton, coupling with calcium signaling. So she can start figuring out now all sorts of connectivity between different types of function. So this is the idea of community detection. In cliques, you can look at communities and you can look at interactions between communities. You can look at how they influence each other. So we can, if you are doing drug targeting, you can start asking the question, how does the targeting influence the interaction between one part of community and another community? Again, she looked at these complex networks from these clique formation from these cliques, and then she looked at very interesting set of studies. This is published, so therefore you can look at it. I'm not going to go into details here. She also worked on developing two new methods. She worked on a method called variational Bayes approach. Again, this is published. And she worked on a method which is called using a very simple idea of Newtonian framework, a Newton's framework for community detection. She used the idea that planets or planetary system or the solar, the stellar system is just simply based on interactions between things. So she used that idea to form, how do you form cliques? For example, how do you know we have the Andromeda galaxy or the Milky Way galaxy? So she used this idea of gravitation interaction approach towards building a Newton type algorithm in community detection. It turned out to be very interesting, and it seems to have the right kind of features. She built all of the graph features and showed that all the standard graph features that you can predict can be obtained from these kinds of approaches. So I just wanted to, I'm going to summarize and let you ask me questions. So I want to conclude by telling you that, there are many, many challenges in life sciences, in biology, which can be approached by information to data to knowledge, data to information to knowledge. In fact, semantics and context are very important in biological data and networks. In my lab, we have developed novel clique finding methods in the context of biology, reducing data into mechanisms, identifying alterations between normal states and disease states and so forth, which involve space, semantics, and structure. So in fact, we bring the concept of space, semantic, and structure into life science approaches. We show how algebraic methods can be used to reconstruct networks in linear and nonlinear biological systems. I showed you about the lasso method, the modified versions of lasso method. You can really look at they involve space and structure. Structure means topology of the network. Space, it involves the space of interactions and the space of the network structure. We use information theory to construct intracellular networks, space, structure, and context. Context is everything in biology. I'm very fond of saying context-free biology is content-free biology. So context is very, very important. So you can use information theory to aid contextually relevant biological networks. And we are developing methods to go beyond correlation to identify causal networks. In fact, we are hoping that we'll use post-Shannon approaches in terms of building causality, in terms of using time series data, longitudinal data to build causal structure and causal graphs and causal networks and so forth. So this is kind of where we are at this point in time. We are hoping in the next, Anant will talk more about some of these issues. In the next 5 years, what we would like to do, if we get supported and funded by NSF, is that we'll be cliques towards building networks from more complex situations. I told you in the beginning about our $120 million project. We want to be able to take the genomes, pediatric genomes, longitudinally, because all these children come from birth to adolescence to this hospital, about 95% of children in larger San Diego area. So we want to be able to sequence them longitudinally so we can look at their lifestyle, we can look at genetic effects, we can look at changes in these, function, physiological function as a function of time, so we can build these types of networks to understand the human condition. So this is where we want to take information theory to.